We all live on planet Earth, a planet where species depend on each other, but a place where especially insects are disappearing from our ecosystems. Research shows a global decline in insect communities. Is this an effect of a growing human impact? As a result of a changing climate? Could we, by tracking the insects, understand our own human footprint? As engineering students, we wanted to help the best we could. So we went to a place we knew had many leading experts, Aarhus University, where we talked to a real-life bug catcher and insect expert, Toge Thomas Høje.
Yeah, so we collect about 2,000 insect samples each season, each summer, and I think roughly we can say it takes about an hour to process one of these samples, and that is to just sort them into broad taxonomic groups. The subsequent identification of the insects down to species level can take anything from yet another hour to maybe even 10 or 20 hours for one sample. And typically this means that we only identify a subset of the specimens that we collect. So an automatic system for identification of insects more broadly would be a game changer for that kind of diversity studies. I think it would allow people that had limited expertise in this area to start considering actually going into this kind of work, and it would certainly allow those people with expertise to speed up the process of processing their sample.
As Toge suggested, we needed an automated solution to handle and sort the insects. We went straight to work. We were able to configure a setup that included a National Instruments LabVIEW controlled universal robot, multiple tools, and vision acquisition. Using the NI Vision Assistant, we were able to find and locate the insects in 6 simple steps. The large insects are handled by a modified tweezer attached to the end of a servoelectric gripper. And the small insects are handled by a suction tool attached to an end effector. When insects have been recognized, they are placed in test tubes for later storage. To summarize, we were able to use LabVIEW to control a universal robot UR5 and multiple tools for the system to run autonomously through a loop in LabVIEW. We were also able to show that even the most delicate processes can be automated. And we also made proof of concept of our initial problem. We would like to thank our advisor Claus Melvad and Toge Thomas Høje, Senior Scientist at the Faculty of Bioscience, for assistance during the project.